Hi students, good evening. Am I audible? Yes, good evening Aditi, Shana, Kirtana, Vignesh. I'm audible? Yes, thank you for that feedback, ma. Yes, good evening Aditi. Yes, thank you every children, thank you so much. So welcome to our new course today, the bridge course, Cell, the Unit of Life. So this is going to be today a very interesting, calm and interactive session, students. So the next whole hour, I'm going to catch your hands and we are going to enter into the cell. Okay, let us start a journey into the cell. First and foremost, before starting the class, let me tell you, you all are the students of science. Yes. So as you would have been studying science, it would be more on to a surface. Like you wouldn't have gone into the depth or the knowledge of what we understand or assimilate the subject. So this bridge course would probably help you to have a better understanding of how you have to perceive the subject of biology, and later how this would help you in your future, as it is going to be like a bridge laying down the various foundation, so that will help you to build knowledge in various aspects of biology. Okay, I assure this today's class will be interesting, and same time it will be a very nice interactive session. So students, yes, come on, we are going to start today's class. Okay, first coming on for the subject of biology, has proudly to say that you're all the future budding biologists. Yes, I hope I can very well use this welcome budding biologist. Yes, can I put the right term here? Yes. So I welcome one and all the budding biologists of the future. Okay, so I welcome you all. So in today's class, today we are going to see a very nice, interesting good evening Rifa, Kirtana, Yadunandan. So it's nice to see you all and hear your names and all that. Welcome students. So today's class I'm going to start with cell. When I say in biology, the first and foremost to start with, to give you a nice idea, a well treat into the minds of the budding biologist, will be the topic of cell. So to start with this topic, because cell, as we all know, it is the basic unit of life. It is the structural and the functional unit of all the living organism. So before I start with any topic of biology for my students, I always speak to them a concept called homeostasis. What you all understand by the term of homeostasis? It is nothing but it is the maintenance of a constant internal environment. What do you mean by internal environment? As we all know, our body is composed of trillions of cells, right? We have a beautiful plasma membrane, a cytoplasm, and then the nucleus. Like that, we have trillions of cells. Okay, inside the cell we call this as the intracellular fluid, and outside the cell is this constitute the extracellular fluid. Now all the cells are constantly bathed by the extracellular fluid, and so I mean the internal environment as the extracellular fluid. Now all the cells in my body are constantly bathed by this extracellular fluid, which is going to provide the medium for it to do all the processes of the cell. Say the metabolism, its various processes or activities. So everything, it has to be maintained in the normal range in the extracellular fluid, and there comes the role of homeostasis. So homeostasis is nothing but the maintenance of constant internal environment. What I mean by the extracellular fluid. Yes. So now when I speak with, we all know that the cells, we have so much of cells, and all these cells which group up in structure and function, they form what we call the tissues. I hope you all know. Now the tissues which all with function and structure, when they have that similarity, when they group up, they are going to form what we call the organ. And this organ, when they group up with, they form the organ system, and as the organ system, all will make up an organism. So this is going to be a very fundamental, and as a student of biology, we all know this, right? Yes, Kirtana, you gave a right answer. Yes, dear, it is the balanced state. Very nice to hear that. So all the cells, tissues, organ comes up the organ system, and put all the organ system comes there an organism. So that is where the subject comes here with biology. So now I'm going to start you from the basic. I said if I'm going to just directly go talk about the organism, the various processes, it will be like Greek and Latin for you all. So I just wanted to start with the concept of cell, which you will be aware, but we are going to give you some more informative data that is going to help you to add on the blocks of already existing idea about the cell, and that is how we are going to learn today about the cell. Yes, is that clear, children? So why did I use the word of homeostasis? It is nothing but because I wanted to tell you, for a cell to function, it should be in a balanced state, that is the homeostasis, and that is how all plays, and that's how the entire organism, as we are being here. Yes, we are being here because all our body is in a homeostatic way. They do perform various actions, and that is what I wanted to tell you all, right? So today with that, we'll start with cells. So now look here, what are you seeing here? Yes, you see those bricks, you see those cement which is being made, and somebody is building their brick block. Yes, is that same thing as how you see every basic block here? Is this the same? The cells are the basic unit of life, and they form the structural and the functional unit of any living organism. This is the concept that you all know. So all organisms, they are composed of cells. Some will have only just a single cell, as you observe in amoeba, or it can be like us where we are all multicellular, right? So it can be a unicellular organism, or it can be multicellular having many cells like us. Okay, so this you have to know. Now coming for the basic of defining a cell. When somebody asks you a definition, I hope you all will give an answer saying, ma'am, it's a basic unit of life, or you will put forth the answer telling like it's a basic structural unit or a functional unit of any living organism. But as it comes on, a unicellular organisms are capable of independent existence, and they perform the essential functions of life. So when you take, say for example, we have a unicellular cell and we have a multicellular cells. So here a unicellular cell is quite independent, and it is doing all the things that is needed for a life, say digestion, respiration, circulation, whatever. But same way when you take a multicellular, yes, even it has everything in it. So what is that which makes up? So anything less than a complete structure of cell does not ensure an independent living. So cell is a fundamental structural functional unit of all living organism. So as you all see here, yes, so what you see that something is moving, like you see that the basic cell, which is the fundamental structural and functional unit of all the living organism. So as you see, everything they are of same size and shape? No. So the cells will vary with their shape, their size, their activities, etc. Right? So now look into this. As I put forth the statement before that the cells are not of uniform size or shape. Now look here, what do you observe here? The onion cells under a microscope. Yes, you see the cell wall that is being bounded and you see a clear nucleus, the cytoplasm, right? You see that. But what about here? The human buccal, where you take a swab and you take a streak and you see here. So the cheek epithelial cells, right? You see a nucleus, cytoplasm. Yes, it has got an outer defining boundary. So what is that? So whatever of any kind, it is a basic giving you a structure and a functional unit of any living organism. So before that, always it is something nice not to forget the pioneers who did a lot of contribution in the field of cytology. The study of cell and its various aspects and its processing function, we refer to a separate branch in the field of biology, what we call it as cytology. So I hope this part we have to emphasize because sometimes they are very much useful, like in examination perspective, as well it is very nice to know certain pioneers to read about them. So we have here a famous person, I hope you all know, it's none other than Robert Hooke. He is an English microscopist. He is credited for discovering the cells. Yes, Robert Hooke, as you see here, he took the plant cells, that is the dead cell walls from a cork, and just he made it appear under the microscope. Imagine in those days with that less advancement of those science and things, people were able to make up of so many things. And just children see now with today with a lot of advancements, like we are somewhere and you are somewhere, and but still we are able to meet each other where we are having a platform for discussing and accessing the knowledge. But in spite of that, okay, we have a lot of advancements, we are still lagging behind the area that in biology, as every student should aspire, is the area of research. I hope students now you will really get the aspiring mind from day now that you also should develop a mind of area where the research. Yes. So far so good. Am I clear? Audible students? You're comfortable? Yes. Like Aditi, Rifa, Kirtana and others, all others. Yes. Vignesh, Sana. Yes. Is that fine? You're able to be comfortable. It's compatible. You're able to follow, right? Okay, so I'll just proceed thinking it's fine. So we have another pioneer, Antonin von Leeuwenhoek. He's a Dutchman. He was the first person to observe and describe a live cell. So probably they say he was the first person to even find the microscope. Robert Brown, okay, he is a Scottish botanist, and the credit for him, he discovered the nucleus, particularly in the orchids. And cell membrane was first discovered by Squan and cytoplasm by Strasburger. So like that we can keep making a lot of people making contribution. We have so much, but only the main things we have highlighted here. You can see the phase contrast microscope. Students, we have microscope, which is because these are all very microscopic tiny particles, some are not visible to the naked eye. So you have to take a cell, stain it up, put it under the microscope where you can see. You have students microscope, you have binocular, we have so many varieties. We have electron microscope. Like that, one such is the phase contrast microscope. The biggest difference with other microscope and a phase contrast was like you can observe a living cell with their internal process. Imagine like you're viewing a cell and you're seeing those processes live, how it would look, right? So that is where the phase contrast microscope gets a credit, and it was developed by Zernike. Okay, I hope now you got an idea of what a cell is and certain pioneers, how do they contributed to the field of the cytology. Now coming to another basic thing here, which I wanted to emphasize you all here is, ma'am, why did you speak about that area of these scientists and these pioneers and all that? I would like to bring you a small example, a small interesting pinch of air. There was one person by name Best and Banting. I hope if you know about insulin, they were the pioneer who discovered the insulin. Now today millions of diabetic people are surviving because of that wonderful thing which they made was the discovery of insulin. So Best and Banting. Banting was actually the professor who was very much interested in setting up a lab, and he was doing on, and Best was a first year medical student. But still he was just a budding doctor, but he was so much interested in the field that along with this professor, he was able to find the insulin and the cause that leads to diabetes, and he was awarded the Nobel Prize. Same way, students, there is no such boundaries or anything refining us. So always any student, as you all budding biologists, when you have that mind and idea of making things, you can very well, you can flourish yourself in the field of biology. Okay, with that, I'm going to now welcome future researchers, future budding biologists, and now we will start with the topic of cell theory. Now, cell theory, when I say theory, it was an hypothesis which was put forth by two pioneers called Schleiden and Squan. It was in 1838, there was Matthias Schleiden. He was a German botanist. He actually had a habit of just examining a number of plants, and he found that every different kinds of cells all that forms the tissues of the plant. So what he found, it was not only a single cell that is making the entire plant. So he saw there are a lot of cells, and these cells that all make up the various tissues of the plant. Like you say a xylem or a phloem or whatever that is there with the cell. So he was astonished with it. Then came another pioneer by the name Theodor Squan. He is a British zoologist, and here he took a pioneer thing in examining a lot of animal cells. So he studied the different types of animal cells, and he reported that the cells has got an outer layer which is called the plasma membrane. I hope you all know that the cell which has got the outer membrane, what we call it as the plasma membrane. To put it in a thing, it's a lipid bilayer. It looks like this. Yes, this is the plasma membrane. Okay, so that was actually reported by the Theodor Squan. So it was Squan and the Schleiden. They were the pioneer to put forth the cell theory. So this is something new that you're learning for the day. Yes. So Squan also said that based on the studies on the plant tissues, that the presence of cell wall is a unique character of the plant cell. So how he concluded that the animal cell had a plasma membrane, here they also said that the plant cell has also got a cell wall. So that was a conclusion he made from his studies. Okay. So they wanted to state that the bodies, so what was the ultimate thing that they stated is, yes, they stated that the bodies of all organisms are made up of cells. Okay, they are made up of cells and their products. Okay, so cells are nothing but both structural and functional unit of the living organism. So you can see that in 1939, Schleiden and Squan, they both together proposed the two tenets of the cell theory. My children, I assure here, what cell theory you are learning now, even when you enter your medical board, MBBS or any stream of medicine or any stream of your biology, this theory is going to come throughout. So just know what are the two tenets that they've proposed. All organisms are composed of one or more cells and products of cells, which I told earlier. The cell is the basic unit of structure, function, and organization of all organisms. The cell is the basic. So it is going to give you the structure. And when there is a structure, definitely it is going to perform an action, the function. So all that group together, they have an organization, and that's how it makes the entire organism. What is the drawback of cell theory? They were not able to explain how new cells are formed. Okay, so it was none other than Rudolf Virchow. He was another pioneer who modified the cell theory by adding a third point into it, where cells can arise from the pre-existing cell. So that was a term that was given by Rudolf Virchow, where he said that new cells are formed from already pre-existing cells. So he used a word what is called the omnis cellula e cellula, which means the cells do arise from the pre-existing cell. That is the statement given by Rudolf Virchow. So when I speak about cell theories, children remember It was Scleeden and Squan who proposed the two tenets, and it was none other than the Rudolf Virchow who stated the third point, telling that the cells are going to arise by division from a pre-existing cell. Hominis cellulae acellulae. Okay. So Rudolf Virchow first explained that cell divided and new cells are formed from the pre-existing cells Hominis cellulae acellulae. So Rudolf Virchow gets the credit by modifying the cell theory by adding a third tenet, that is, cells will come from the pre-existing cell. Yes, is that clear? So shall we go to the question and answer session? Yes, I am waiting for your answers. Cell theory was given by Antonin von Leeuwenhoek, Robert Hooke, Scleeden and Squan, Robert Brown and Rudolf Virchow. Yes, please answer the question. I am waiting for your answers. Yes, students, please post the answers. Rifa, Aditi, Kirtana, Vignesh, Sana, others, please. Yadu, Nandan. Yes, that's great. Everybody are giving your right answers. I'm happy. Yes, even if it's right or wrong, it's not a matter. Once when you are entered into a platform of learning, faults are common. Only when you make errors and mistakes, you can learn in life. Learn this from me. Yes? Yes, that's nice. Everybody are giving the right answer. A big hand of applause. Super students. Cell theory was given by Scleeden and Squan. Right? Shall we go to the next part? Yes. Now we are going to the next question. The statement Hominis cellulae acellulae, which means all cells arise from pre-existing cells, was given by Rudolf Virchow, Scleeden, Robert Brown, Antonin von Leeuwenhoek. Yes, I hope you will be knowing the answer. Yes, please post the answer. Yes. Yes, I'm seeing here the answers coming up. Please look into the statement Hominis cellulae acellulae. Yes, 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 all are bombarding with answers. Budding biologists, you people are awesome. You're just giving with all nice colorful answers as I seeing in your page. Yes, very good. The answer is right. It's none other than Rudolf Virchow. So is that clear? This is how you have to build the concept or the knowledge of biology as we slowly ascend into one another. Now we'll come here about the cells overview. I hope in schools, in your middle school and all that, you would have learned about cell, what is prokaryotes, eukaryotes and all that. But still, some of the basic things like you wouldn't have understood it really, or you would have missed it actually. But as when you want yourself to become a future biologist, certain things you need to have a clear-cut concept, and that is what we are going to make today with cells overview. So look into the cell shapes. So cells may also be shaped to be polygonal, right? They can be cuboidal, columnar, concave, cylindrical, disc-like, thread-like, long, irregular. Man, what's all? Like they are of so many shapes. Like you can see the cuboidal cells that is lining the epithelial cells of thyroid gland, the columnar cells that is lining the epithelium cells of your stomach, the biconcave cells of your RBC, right? Those WBCs, they are mostly like they don't have a perfect shape, but they are amoeboid. Like I said, the columnar epithelium, sometimes they may be like thread-like, they'll be long, irregular or disc-shaped cells. Cell shape may vary, but the shape of the cell, that depends on thing. So for that, let me give you the idea with RBC. Have you ever tasted that sweet badusha? I think mostly kids of this generation wouldn't have. You would have all gone for that kaju katli and other nice milk sweets and all. It's one of the traditional sweet badusha. And how you take, next time when you go into the sweet shop after all this lockdown and all, just ask how a badusha would, or look into a Google and see. The badusha, how you see, will be the same electronic microscopic view of RBC. It's a biconcave disc-shaped structure. Now this RBC, it's biconcave. Can anybody tell like this biconcave shape is for what? Mainly to perform that specific function of transfer of or transport of the respiratory gases. So the shape is that one which is confer or offering the function of a particular cell. Same way here, the shape of a cell, it depends on the surface tension, the viscosity of the cytoplasm, the mechanical force that is exerted by the adjoining cell, the membrane rigidity, as well as how a cell is adapted to function. Like unless if the RBCs are not biconcave, they cannot squeeze through the tiny capillaries, very, very minute capillaries. If they are not biconcave, they cannot squeeze through. If they are going to be somewhere, say a big oval shape, they will get stuck up, and in that, as they want to get out, they'll get ruptured. So to avoid all this, every cell do have a specific shape for it to perform a specific function, and to have the cell shape, it has got a lot of factors like the surface tension, the viscosity, the mechanical forces, all that do make and gives that. Now when I say the shape of RBC, probably the biconcave shape, it's actually given by a protein called spectrin and ankyrin. Like that, you have a lot of membrane proteins and all that is trying to pull it and make it in that shape. Okay, so that is where is it. Vignesh, you asked me a doubt about there will be layers, ma'am. Can you ask me like what question so that I could answer you, please? What are the layers that you're talking about? I can explain you, ma. So here, what you observe here, you see the neuron, which is a structural and functional unit of nervous system. So you can see the neuron, right? This is the cell body. Okay, these are the short process or the dendrites as we see. These are the long process or the axon as you see. So you can see the trachea. Ma'am, I was saying about that sweet. Oh, so sweet. You're asking about that badusha. No, I think it has that layers. Yeah, you are right only. Yes, it has layers and it will be biconcave shape. Okay, so next time we will show that picture of when we are going for RBC cell. I will put that picture for you. Good. And I was so wondering what layers. I really thought honestly that you are asking about the epithelial layers. But so sweet that you asked about that sweet. Nice. Okay, so you have a trachea. See how elongated a cell could be. Or you see the round oval, those mesophyll cells that we find in the, what you call the leaf of that plant cell, right? So these are various cell shapes. Now this, see, it's a quite branched and long, quite irregular also, right? So now when I look into the size of the cell, my God, from where to where we are. Yes, see, ostrich is 120 mm. Yes, human egg, see, 0.1 mm. And look into the RBC, it's 7.2 micrometer in diameter. Look at the RBCs. Yes, the monocytes are the largest. They are around some 18 to 22 micrometer in diameter. And see, we have those lipid molecules, the PPV yellow, that is the mycoplasmas what we are talking about. They are supposed to be the smallest cell. They are only 0.3 micrometer in length. And the largest isolated single cell, as you see the ostrich. Okay. So the nerve cells are the longest as we saw. Yes. So that is somewhere you imagine, no? Like what I suppose something is coming on, but still the impulse is going on. So you can imagine such long nerves are there, branching like arborizations of a tree, branching like a tree branches. They are longest cells. So mycoplasma, smallest cell. And we have the largest, as you see, the ostrich. Human egg is only 0.1 mm. Bacteria, see, it's 0.7 micrometer. But here it's in mm. Okay. So that you need to appreciate it. So coming off of the types of cells, we have eukaryotic cells and prokaryotic cells. So coming to the eukaryotic cells, actually to speak on, no, I usually teach my students telling types of cells like there will be undifferentiated cells and differentiated cells. When I say undifferentiated cells, I am talking about the stem cell. Stem cells are cells that has got the capability to, or a potential to transform them into any type of cell. But when I say differentiated, they are the post-meiotic cell. They are very specialized. They know what to form. So they are very, very determined and what you call, they form that specific type of cell what is. But coming here, as in all textbooks of biology do they give, they say types of cells. So eukaryotic cells and prokaryotic cells. So when I say eukaryotic cells and prokaryotic cells, what are we trying to talk about? So it is nothing but, here they have given the examples like protists, plants, animals and fungi. But what about the prokaryotic cells? We include all the bacteria, we include the blue-green algae, the mycoplasma, that is the smallest one which I said, the PPLO, that is the pleuropneumonia-like organisms. So they are all prokaryotes. What are prokaryotes and what are eukaryotes? When I say prokaryotes, here all they are the cell, actually they are not bounded by the double membrane. So they are all not bounded with the double layered membrane. That is where a prokaryote is. So you can say that that is in prokaryote, it will all the organelles mostly they might be naked, like they'll not have a envelope covering within it. And coming on for the size of a cell, they'll be very small here, whereas it is comparatively large. Okay. And you can always say that the prokaryotes, they have a very incipient nucleus in the form of nucleoid. They don't have a proper membrane bound cellular organelles, whereas a eukaryote, you see they have a proper membrane bound organelles, yes, and they have what you call an advanced nucleus, and they have all the true nucleus in them. Here you don't find such true nucleus here. Okay, so that is where another difference comes. See, you have a cell wall, and sometimes in some prokaryotes you can see what you call as a capsule. Probably in eukaryotes cell wall might be there. You have the nucleoid, what we say here, and you have the flagellum and certain sort of pili like that, or Fimbriae and all. But you can see the cell membrane. So you can see the outer the cell membrane. Okay, that double wall envelope will be there, and it will have a true nucleus with all the membrane bound cellular organelles and all. So that is where the difference. So you can see the organelles like Golgi apparatus, mitochondria, the cytoplasm, the jelly like substance in which the organelles, you can really imagine, like I'm all taking your hands, just close your eyes and imagine that you're just putting your leg into it. That's a plasma membrane. We are just entering, there's a jelly. All are shouting, ma'am, something is looking like very jelly like. Yeah, that jelly is where you come and touch. You're seeing the mitochondria there, you're seeing the Golgi apparatus, there comes this endoplasmic reticulum, and you're seeing the beautiful true nucleus which has got the controlling, the head of controlling the entire cell activity. Okay. Yes, now coming for the question session. Nuclear membrane is absent in Plantae, Protista, Monera, fungi. Yes, ma'am, in RBC, the spectrum ankyrin makes up the cytoplasm or the plasma membrane? It makes up the membrane. It makes up the membrane. I'll explain you this. Now answer this question, Kirtana. Kirtana, you answer my question. Then I will answer your question. Yes, come on. Kirtana has asked a nice question. She's saying, ma'am, you said spectrum, ankyrin and all. Ma'am, where are they? Nice question. I'll explain. Please answer this question, children. I'm waiting for your answers. Yes, come on. Budding biologists, please post your answers. Yes, it's none other than what happened. Please post. Kirtana, Aditi, Rifa, Ria, Rifa, Yadu, Nandan. Yes. Is none other than the Monera. Because just go back. Yes, I already said they are present in the where? The bacteria, the blue-green algae, mycoplasma. So where is that doesn't have? It is the Monera. Yes, very good, children. You're giving the right answer. Kirtana has asked a question, ma'am, in RBC, the spectrum, ankyrin and spectrum, where do they make up the cytoplasm? It makes up in the cell membrane, ma. So that confers the biconcave shape of the RBC. They say that the congenital absence of, congenital from birth, ma, congenital absence of the spectrum, that is in the RBC membrane, it seems it will make the RBC spherical. Okay, and once when it goes to those tiny capillaries, they get ruptured and lead to a characteristic clinical condition called congenital spirochetes. Okay, so they are in the membrane. If they are not there, they become permanently spherical, and that is not fair. It leads to congenital spirochetes. Okay, is that right, Kirtana? The smallest cell of 0.3 micrometer in length is ostrich egg, cyanobacteria, bacteria, mycoplasma. Please post your answers. Yes, very good. I see people giving up the right answer. What about this question? Yes, please. Yes, you are right. It is present in the mycoplasma, that PPLO which I say, right? Yes, I see you all posting the answers. Yes, very good. Superb. You're coming with answers. Now today for the day of discussion, shall we move on to the prokaryotic cells? Yes. Now coming to the prokaryotic cells to speak with. Okay, I just wanted to show you the size of the prokaryotic cells. Yes. Yes, very good children. You all are giving the right answers as I see in the pic that is you've been making up. So now we're coming to the prokaryotes, mostly the kingdom Monera, which we always speak about. So here when you look into the typical thing, say they are around 10 to 20 micrometer, but see about a typical bacteria 1 to 2 micrometer, yeah, that's around. And the PPLO see very less. In virus see they were still more small. They are around 0.02 to 0.2 micrometer. But look at the hangama they are making with the COVID, right? They are very bad, right? So look into the eukaryote and see what are the various prokaryotes that they are making up. Okay, let me start with today's topic with nice prokaryotic cells. So when I talk about the kingdom of prokaryotes or whatever, I usually speak starting with the Monera. Okay, here the prokaryotic cells, they are represented by bacteria, the blue-green algae, mycoplasma and PPLO. See, they are very smaller. Mostly we say that the average size of bacterial cell is around 2 mm, and they are generally smaller. They multiply very rapidly than the eukaryotic cells. They are very greatly in size, shape, all that. So you can see some of the small forms, like they can be from 0.15 micrometer. Some can be as large as even 600 micrometer. Okay, the size. So when you look into the various shapes, as we all see here in prokaryotic cells, we can see here they are of coccus, the bacillus rod-like, spirochetes, Vibrio, which is comma shape, and the spirilla, which is spiral shape. So when you take this coccus shape, as you all see, the best thing I can give example like Diplococcus. So they are in groups of 2 Diplococcus, or they will be in just occurring singly like how you see a cocci, or they will be in groups of 2 like that. Streptococcus, like they will be in chains, like this in chains, Streptococcus. Staphylococci will be like irregular clusters, like that. There are many type of cocci. When I say bacillus, they will be cylindrical shape, mostly like a rod shape, like streptobacillus, or they can be like what you call or diplobacillus. We have what is called the spirochetes. The best example I can give you will be Treponema pallidum. Okay, so this is the one which is that causes syphilis. So the best example I can give for spirochetes is the Treponema pallidum that causes syphilis, that bacteria. And then we have the Vibrio. It's a comma shaped bacteria. So I can speak about the Vibrio Cholerae, that which induces cholera. I hope you have heard about cholera. It's quite epidemic. It was actually making the whole place into a thing where people will have rice water like loose stools, and there will be severe diarrhea, severe vomiting. They go for total dehydration and circulatory collapse and death. It was such a big thing. Okay, Vibrio Cholerae. Okay, and then we have the Spirochetes, that the thing which is there. So there are various spiral shape also, like as you see the Spirochetes. So like that we have different shapes of these bacteria as we see. Now coming on for the organization of a prokaryotic cell, the first and most fundamental, as my students should understand here, that basically every prokaryote, they have their own shape probably to do the necessary function, that everybody should understand. Now when I take a structure of a prokaryote, it has got a cell wall, okay, and surrounding the cell wall, what it has got? It has got the cell membrane, right? And in that it has got a fluid cytoplasm, which we call it as what you call a cytosol or a cytoplasm. That's a fluid filled, that is filling the cell. So every prokaryote, it will have a cell wall, a plasma membrane or a cell membrane, and then you have a fluid portion, which I said I'm just taking you inside. We are seeing the cytoplasm. It's there in all except the microbioome, because there itself is very small. Where you find the cytoplasm? You don't find them. Okay. Now when you see all these cells, they are all very what you call the cell structures or these cell envelopes, we can actually study only with the help of electron microscope. Just like that naked eye or ordinary microscope will not help us to study. So you have to know that the cell envelope is basically of 3 layers. What are the 3 layers? We have the outermost, which is called the Glycocalyx, then we have the cell wall, and then we have the cell membrane. Okay, so the outermost is what? The Glycocalyx. The Glycocalyx is actually nothing but it is made up of most of the macromolecules. So they are very tough, somewhat sharp, defining layer. Sometimes it can be a loose sheath called a slime layer. So the capsule and slime layer, they will be made up of the polysaccharides. Okay, they will be made up of polysaccharides, and sometimes they may also contain the proteins. Okay, so we'll see what are those functions. So you can see that all these envelope together is what meant for protection. So when I see it in the electron microscope, it's not just a single layer. I'm going to appreciate a 3 layers with a Glycocalyx, a cell wall, and then a cell membrane. The Glycocalyx is of almost of the macromolecules, and all the 3 is going to have a single protective unit. Now I said the Glycocalyx will differ in the composition in the previous slide, said that it has got a slime layer which is very loose, or sometimes they can be thick and tough. That is meant what is called the capsule. Now what are the functions of the slime layer or the capsule? First and foremost, they are going to give us the protection. So they are going to give the protection, and next is that they are going to block the attachment of various bacteriophages. Number 3, they inhibit phagocytosis. Okay, and sometimes they can even prevent or burst from collapsing. Okay, they prevent the bacteria from bursting or collapsing. That is what they meant for the cell wall, the layers, which is so the another important thing is they may prevent loss of water and nutrients also. Okay, so the plasma membrane is next to this. It's semi-permeable, and that is the one which is interacting with the outside world. So the plasma membrane, like any other eukaryotic cell, it will be the same, in which this will not have sterols such as cholesterol. So here the plasma membrane will be same, but they don't have cholesterol. But however, the sterol molecules, like in terms of some hopanoids, they might be present. So coming here for the bacteria, see the bacteria are grouped, the envelope, they are grouped into 2 groups. Depending on the layer, it was Gram, Christian Gram. He was a pioneer who studied the bacteria and labeled them as gram negative and gram positive. Students, I think if you would have done a urine culture or a blood culture, either of yourself or somebody at home, you would have seen these names like gram negative, gram positive and all. What are we talking about? The bacteria, depending on the cell envelope, we are going to, plus the staining procedure developed by Gram, we are going to call them as gram negative or gram positive. When they take up that gram stain, we call them as gram positive, and that which does not take up the stain, we call it as gram negative. Look into this gram positive. They have got a very thick peptidoglycan layer, but they will not have that outer lipid membrane. But in gram negative, you can see a single layer of peptidoglycan as well as a outer lipid layer. So this is where the difference comes. So when I say gram negative, E. coli, Helicobacter bacteria, they are all gram negative. Gram positive, I can say Corynebacterium. Okay. So they are all gram positive bacteria. So next, coming to the interesting structure, what we call the mesosomes. Now the plasma membrane, right? We all know that the plasma membrane will retain the cytoplasm, and it is a selective permeable layer. So plasma membrane is a selectively permeable barrier. So now some of the plasma membrane will have invaginations like this inside the cell, what we call it as mesosome. So the characteristic in a bacterial cell, some of the plasma membrane, they have invagination or infolding, what we call mesosomes. So these mesosomes are nothing but they are involved in cell wall formation, or they will be involved in chromosome replication, or some secretory process. So that is the job of mesosome. So you can see those infoldings into the cell, and these extensions on the form of vesicles, tubules or lamellae. So they help in some replication or cell wall formation, distribution to the daughter cells, or some secretory process like in respiration, secretion process, or increasing the surface area. So suppose you fold the curtain, what will happen? Like the surface area is more, so and the enzymatic content also. So that is the job of mesosomes. Then we have what are called chromatophores. The chromatophores are nothing but they are the internal membrane systems in the prokaryotic cells. So the chromatophores are the internal membrane systems in the prokaryotic cells. That is going to be complex in some photosynthetic bacteria like cyanobacteria or purple bacteria. So here you can see they are aggregates, like you can say there are some membranous extension that what you see, okay, what we call it as the chromatophores. So they contain the pigment like in cyanobacteria or purple bacteria. Yes students, so far you are all able to follow. Any doubts? Am I audible and is fine, right? Now look into this, highs, look into this. This is how when we move into the microscopy, look into this, how beautifully the bacteria, how do they move? Yes, they are moving. Movement is called motility. That's the flagellar motility of the bacteria that do we see. Yes. Now coming here for the motile units here, the bacteria can be motile or non-motile. Probably they say that there is one thin filamentous extension that do come, what we call the flagella. And besides the flagella, we have another thing what is called the pili. And apart from that, we have those structures which is just sprouting out, what we call it as the Fimbriae. So we have the Fimbriae, the pilus and the flagellum. So now we will see what is there. So now the pilus and Fimbriae do not have any role in movement except the flagella. So when you look into the flagella, actually the flagella, it has got three parts. It has got a filament, right? It has got a hook and it has got a basal body. So here when I speak about the flagellum, actually you should know that the flagellum are the structures that here you can see the filament is actually made up of the protein flagellin, okay, and the basal body will be attached to that plasma lemma, and then you can see that the basal body, that whatever that base, no, it can rotate at an angle of 360 degree. So the filament is supposed to be the longest portion and it can extend from cell surface to outside. Okay, so that part you need to know. And I'm going to talk to you all about the pilus. Now when I talk about the pilus, students, I'm audible? Is that clear, ma, for everybody? Any doubts so far? I'm just making you now, taking you into the concept of prokaryotic cells. I'm explaining you about the prokaryotic cells and particularly giving you an overview because you would have studied like, but I'm telling you certain points which you are like mesosomes and what is their role or the motility part of the bacteria, like that. Able to follow? Yes? Okay, fine. So now when you look into the pili, they are actually the elongated structures which you see. So they are actually called the hollow, narrow, protoplasmic outgrowths. In some of the bacteria, these pili, no, we just call them as male cells or donor cells. So they are going to have a major role as sex pili in conjugation. I hope you would have heard about conjugation tubes formation all. So pili do make a very important role. They made up of special protein. So coming to the Fimbriae, they are actually small bristle-like structures, those Fimbriae, that is sprouting out. So they say that helps the bacteria to attach itself to certain rocks or streams or even to the host tissues. Okay, so Fimbriae is something are known to attach them to a solid surface. Okay, so they are responsible for the clinging of the cells to that particular substratum. So that is the role done by the Fimbriae. Okay, so the pili or Fimbriae, they'll be numerous, but when you take they'll be shorter than flagella, but they don't have any role in motility. This basic you should know. Okay. Now coming to the ribosomes, as you all know, the ribosomes, they are non-membrane bound organelles. That is one thing. Both eukaryote and prokaryotes, those ribosomes, no, they are not bounded by membrane. Like how you see a mitochondria or any other structure. So in prokaryotes, the ribosomes, they are associated. So as you see the ribosomes, so they'll be associated with the plasma membrane of the cell. So they'll be varying from 15 nanometer to 20 nanometer. They are basically made up of two subunits. What totally, when I say ribosomes of prokaryotes in the exam, when they ask, you say it is 70S. But when they ask you as a two basic subunits, we say it as 50S and 30S. Ma'am, what are these ribosomes 50S and 30S and all? Like we call it, depending on the sedimentation coefficient, Sweetberg's unit, we mentioned them. So it is 50S and 30S. It's a larger and a smaller subunit. But on the whole, when we represent and together speak, we call it as 70S as prokaryotic ribosomes. So what is the role of ribosomes? Yes, definitely they are going to play a very important role in protein synthesis, right? So in ribosomes, when you see, here several ribosomes, they will all attach themselves, they will form that mRNA, the messenger RNA. So that form the polyribosome or polysome. So that is how the mRNA is going to get translated into a protein. So because to make it very specific or make it very simple, a prokaryote is like a single room now where you are sitting and listening to a class. In a single room is like a prokaryotic cell. But when you look into a eukaryotic, you have a lot of compartmentalization. Like everything is compartmentalized, everything is featured there, everything has got a role. But here it's just a single whole room where everything should take place. And that role of ribosome is protein synthesis, making a polysome, that is giving a translation, and of that mRNA is being translated, and that's how the proteins are being formed. Okay, and I could see here that there are some inclusion bodies, as you see here. Those inclusion bodies, they are nothing but the reserve materials that are stored in the cytoplasm as inclusion bodies. So they are glycogen granules or phosphate granules, sometimes sulfur granules, depending on that we might have. So you can see the nucleoid or that genopore we call. It consists of a single strand of circular DNA that has been repeatedly folded with the help of RNA. So the DNA is naked, it is not associated with histone protein. That is also another difference with eukaryote. When I do eukaryote, you will be able to appreciate of what is things that is becoming more complex. Here things are not there. Okay. And then you can see those inclusion bodies which I spoke just before. They are the reserve material which are stored in the cytoplasm. They are not bound by any membrane system. They are very free, like phosphate granules, synophysin granules or glycogen granules. Okay, like that. And sometimes we see gas vacuoles also, like we will see them in blue, green, purple and green photosynthetic bacteria also. Okay, so we say those volutin granules that stores the phosphate. Okay, so that is where they are they are called like metachromatic granules. They store they take up different colors, that's why that name is there. Okay, then these gas vacuoles, as you all see, you're able to appreciate these gas vacuoles. You can see in the blue green algae, right, some purple green photosynthetic bacteria. So what is the purpose of a gas vacuole? They are going to provide the buoyancy for the bacteria. It's helping in floating. So that is where the job is done. Now coming to the nucleus, as I explained earlier, there is no well-defined nucleus. The genetic material is quite naked, not enveloped by a nuclear material or a membrane. Yes, students, you are able to see. So what is that you need to know? The genetic material of a prokaryotic cell is known as nucleoid. Okay, so the nucleoid is something like a singular strand of DNA duplex that is repeatedly folded by an RNA. So in addition to the genomic, we find that is a circular DNA. Many of the bacteria have small circular DNA outside the genomic DNA, and those are called the plasmids. So some of the, apart from this genomic DNA, we have other smaller DNA, they are called plasmids. So what are plasmids? They are nothing but small extra chromosomal rings of DNA that is present in Munira and other organisms. So they have the capability to replicate, self-replicate, okay, of nucleoid. So that is important. Okay, so that we need to know. So the plasmid DNA confers unique phenotypic characters to some bacteria. So they also possess the character to provide resistance to antibiotics. So in future, when we are going to study about the biotechnology and various applications of these bacteria, like particularly the E. coli in production of various substances, like in production of vaccines or certain drugs and all, how this plasmid is helping us, we'll be doing that in detail. So the plasmid DNA is used to monitor the transformation. So they are non-vital genes only, but they may or may not be useful to the bacteria, but they are also used to modify or monitor, monitor the bacterial transformation with the foreign DNA. There are three types of plasmids. We have F plasmids, R plasmids and Col plasmids. Okay. F is called the fertility plasmids, the fertility factor or the F factor. The R is one, that is they are resistance against some common antibiotics. So resistance. And then we have what is called the Col plasmids. That Col plasmids like very specific with coliform bacteria. So that is where. Okay students, now we'll come for the question session. I hope so far I just gave you an overview with the prokaryotic. With that idea, I'm just going to discuss certain questions. Which of the following cell organelles is non-membrane bound and found in both prokaryotes and eukaryotes? Lysosomes, ribosomes, centrioles and mitochondria. Yes, please post your answer. Yes, Kirtana, Aditi, Rifa, Yadunandan, Vignesh. Please post your answer. I'm waiting for your answers, children. Yes, yes, yes. Very good. Yes, as you all said, yes, it's the right answer. It's the ribosomes, which I already said. The structure in both pro and eukaryotes, they are not bounded by membrane. And they are going to, yes, it's right. Vignesh, you not need to put a query, you're right. So it's mainly meant with protein synthesis. Another important thing to carry for the day, it is 70S. Okay, good children, very good. The subunits of ribosomes of prokaryotic cell are 60S, 40S, 20S, 90S, 50S, 30S, 30S and 60S. What should be your answer? Yes, all are giving the right answer. Very good. Yes, please post the answer. Yes, very good. The answer is right. It is 50 and 30S. Yes, very good. The genome DNA of a bacterium is circular. Good. Linear, segmented, rod shape. I see all of you are giving the right answers. Excellent children, excellent. Now just see the genomic DNA I'm asking about. What is the shape? Please listen, I'm asking about the genomic DNA. Genomic DNA, children. Yes, it's right. It is circular. It's not segmented, Rifa on Kirtana. Okay, the genomic DNA which I showed you, you had that what you call the what I said, the genopore or the nucleoid. They'll be in circular. Okay, circular fold. Okay, the duplex, single strand. So they are circular in nature, not segmented. Okay, understood, Rifa and Kirtana? Which of the following is not a structure of prokaryotic flagella? Filament, centriole, hook, basal body. I spoke about the structure of flagella. Which is not the structure of prokaryotic flagella? Yes? Yes, please, Rifa, Kirtana. Very good. Come on, give the answers. Vignesh, Sana, please post the answer. Yes, you are right. What did I not say? I said it has got a filament. Yes, it has got a basal body and it has got a hook. Right? It has got a basal body here. Okay, so that is where it doesn't have that centriole. Yes, very good. Yes, everybody is superb. The dash are the small bristle-like fibers sprouting out of the bacterial cell. Pili, mesosomes, cilia and fimbriae. What should be the answer? Yes, very good. Vignesh, Kirtana, Aditi, Rifa, super. Just answer this question. The dash are the small bristle-like fibers sprouting out of the bacterial cell, pili, mesosomes, ciliae, fimbriae. What should be your answer? Yes. You can put the numbers, ma. You can put the numbers, that will be fine. Yes, you are right. Very good, Kirtana. The answer is fimbriae. So you can see that bristle-like fibers, like I said, that helps in clinging of them to the surface, right? So that is the right answer. Yes, very good. Sana, Kirtana and all that. Good. Yes, children. Good, Vignesh. Yes. So with this, I today made you en route into the cell, gave you an overview of cell and particularly the prokaryotic cell. Hope you are comfortable, you understood. Any queries? Any doubts? Budding biologist, any doubts? Shall we wind up the session? Is the session interesting? You're able to follow? We'll catch up you with the next sessions. Okay? Thank you, students. Bye. Good night. So thank you, students. Bye. Good night. GG.